Have you ever been on a really boring date? Like the kind where you're just sitting there while someone else is talking and you're reading the like sweet and low package to entertain yourself? And maybe you've seen on that artificial sweetener that it says something like shown to be a potential carcinogen in the state of California. And you're wondering to yourself, like, number one, how can something be a potential carcinogen? Like, we're not really sure. Does it cause cancer or doesn't it cause cancer? And then why in the state of California? Well, this brings us to the Ames test. The Ames test was invented by a very sexy-minded 1970s researcher in California, Bruce Ames, and he really realized that we could utilize a bacterial system to help us determine if some chemical was likely to cause cancer. And the way that he did this was to look to see how likely it was that that chemical would cause mutation in a bacterial system. And he used a particular bacterial system. He used an auxotrophic mutant of Salmonella. So remember talking about auxotrophs. These are the mutants that lack the ability to do something. They lack the ability to biosynthesize a particular growth factor. In this case, the amino acid histidine, my favorite. So the auxotrophic mutant could not make histidine, could not grow on a medium that lacked histidine. So what he looked for is that if a chemical was mutagenic enough, it would cause reversion, noteworthy levels of reversion of a histidine auxotroph backwards to being prototrophic. So it would recapture this mutant phenotype. It would cause the histidine auxotroph to become prototrophic. It would be able to live now in the absence of added histidine. So here's how the Ames test works. You begin with this histidine auxotroph of Salmonella in a test tube, and you have two medias on which you will plate it. So you have a rich medium with a small amount of histidine, that's the control. And then you have a rich medium with the suspected mutagen and a small amount of histidine. So we know that this suspected mutagen, we're thinking it's going to cause reversion. It's going to cause the histidine auxotroph to become a prototroph. But why the small amount of histidine? I mean, notice in the control and in the experimental, we're adding a little bit of histidine. Well, why is that necessary? Well, remember that in order to see mutation, you actually have to have replication. So a little bit of histidine is required to enable this auxotroph to replicate. So there's got to be a little bit there. And then we can see, hey, to what level is mutation taking place to enable revertance to form? And we would hypothesize, right, that this mutagen would allow for a greater level of reversion than we would see spontaneously in the control. So in this particular experiment, we can see that there are a certain number of spontaneous mutants. We expect that, right? 1 in every 10 million to 10 billion, we might see a spontaneous mutation taking place, but we would expect the added mutagen to cause that to be much higher, and we would see more reversion. So here you can see a greater number of colonies forming in the experimental plate where the suspected mutagen was added. Now, this isn't always going to catch all mutagens. We're not going to see 100%. And so this whole idea of saying, well, potential carcinogen is one way that we can label that to be safe. Additionally, sometimes we don't see a mutagen acting like a mutagen until it's been processed by the body. In many cases, particular processing within the liver may cause a chemical to go from being not a mutagen to being a mutagen. So sometimes it's actually common to put a liver extract also in here to see if it's the conversion, the processing of a chemical that is actually what makes it mutagenic. So this test is an extremely valuable test, and I want to look with you at an article, at an abstract for an article that's diving into the chemicals that are utilized in the textiles industry. And in case that sexy-minded term is not familiar to you, the textiles industry is the clothes-making industry. So a lot of dyes are utilized in making our clothes, and a lot of our clothes are made in places where there aren't regulations in place to stop the use of a dye that might be carcinogenic. So let's look at this abstract. So this is a 2017 publication. The combination of chemical analyses and bioassays allows the identification of potentially mutagenic compounds in different types of samples. There we go, right? Potentially mutagenic compounds could be detected by the Ames test. Dyes can be considered as emergent contaminants and were detected in waters under the influence of textile activities. That means you've got a factory making clothes. They're using these dyes and they're sending them downstream in the river. So the objective of this study was to evaluate the contribution of 9 azo dyes to the mutagenicity of representative environmental samples. Samples were collected along 1 year in the largest conglomerate of textile industries in Brazil. We analyzed water samples from an important water body, Piracicaba River, upstream and downstream of the two main discharges, the effluent of the wastewater treatment plant, so WWTP, remember working with that in lab. Actually, you probably haven't gotten to that lab, wastewater treatment plant water that is coming downstream. And the tributary to the Quilombo River, which receives untreated water from local industries. Samples were analyzed and tested for mutagenicity, and we can see different forms of analyzing for the presence of dyes, and then testing them in this salmonella system, so the oxytrophic mutant system. And so we can see these 6 dyes were detected, so via the LCM, SNMS, and these were the dyes that we see being detected. And then the most sensitive condition for the condition for the detection of mutagenicity was one particular strain of salmonella. The concentration of dyes and the mutagenicity levels varied along time, and in the dry season, right, it becomes more concentrated in the dry season in that wastewater. So dispersed blue 373 and dispersed violet 93 were the major contributors to mutagenicity. So these were what caused the greatest degree of reversion in the oxytrophic mutant. And so we're recognizing that water quality is severely compromised by sending these downstream. So, you know, this is why you wonder, Rachel, why does she only buy her clothes at thrift stores? This is why. I don't like to feed that industry. So this is a really interesting example of a use of the Ames test, and definitely worth flexing your sexy mind around.